everyone, welcome to the session of concept of enzyme regulation. When the body is in a well-fed state, certain enzymes are switched on, other enzymes are switched off. At the same time, when we are slowly creeping into the stages of fasting, whatever is off is now on, the others are switched off. Who is regulating these enzymes? So that is what we are going to learn, the concept of enzyme regulation. One more thing, this is the cornerstone of every metabolic pathways. If we don't have a proper understanding of this topic, then the biochemistry will be a nightmare for you. So we can make this topic very easy. So that is what we are going to discuss now. Let's dive into the topic. So the first thing is that when the body is in a fasting stage, what is shown here is the plasma membrane. In the plasma membrane, you can find a receptor. The name of this receptor is G protein coupled receptor. Okay, this is a transmembrane receptor and you can see a guy which is attached to it. It is a G protein. That poor guy G protein is very sad now, waiting for its ligand. Now, in another way, we are saying that he is waiting for his friend to come. In biochemically, it is ligand. Okay, so which are the ligand for this receptor? The ligands can be glucagon. Okay, another ligand is epinephrine. So glucagon and epinephrine are the ligands for this receptor. And even though I have shown both glucagon and epinephrine here, doesn't mean that both glucagon and epinephrine has to bind. Either glucagon or epinephrine can bind to this receptor. Where do you find glucagon? The glucagon is seen only in the liver. Then what about epinephrine? Epinephrine can be seen either in the liver or in the muscle, which means in muscle you will find only one friend for this G protein coupled receptor, and that is epinephrine. And here I just told you about a fasting stage. But remember, epinephrine is otherwise called as adrenaline. So when adrenaline rush is coming, then also the same cascade pathway is acting. That is during the fight and the flight reaction. Epinephrine has a major role. Okay, so now what we are going to see is when glucagon or epinephrine binds to this G protein coupled receptor, we can see the G protein is active now. This G protein is going and binding to another enzyme which is adenylyl cyclase. Now when the G protein binds, you can see that the adenylyl cyclase is active. What's the action of this adenylyl cyclase? It is going to act on another biomolecule, that is our energy currency, which is adenosine triphosphate. Now this adenosine triphosphate is converted to a compound which is called cyclic AMP. What do you mean by cyclic AMP? In ATP there are 3 phosphate. In adenosine monophosphate there is only 1 phosphate, which means when ATP is converted to cyclic AMP, 2 phosphates are removed. Not only that, this ATP is cyclized and hence it is called cyclic AMP. Okay. Now there is somebody who is waiting for the cyclic AMP to come, and that is our cyclic AMP dependent protein kinase. Now this cyclic AMP go and bind to the cyclic AMP dependent protein kinase. Now what happens? The cyclic AMP dependent protein kinase is active. What's the action of kinase? Transfer of phosphate. Okay. So, there are some enzymes, regulatory enzymes in our body which are getting phosphorylated, which means enzymes are getting converted to a phosphorylated state. What do you mean by this enzyme getting phosphorylated? We know that enzymes are proteins, which means they are made up of amino acids. Now, this phosphorylation is happening to one or other amino acid. Specifically, which amino acid? The hydroxyl group containing amino acids are getting phosphorylated, especially serine. That is the most common amino acid that is getting phosphorylated. The second is threonine. And the third possible is the tyrosine. Okay, so these hydroxyl group containing amino acids are getting phosphorylated, means the enzymes are getting phosphorylated, which means when the body is fasting, number 2 situation is when the body is in a fight or a flight reaction or a stressful condition where there is adrenaline rush, the enzymes are in which state? They are in phosphorylated state. Now for this phosphorylation, what is the other name for it? It is called covalent modification, which means these hormones are regulating the covalent modification. That is why we use the word hormonal regulation or covalent modification. Both we can use as a method of enzyme regulation. So the summary here is that in the stage of fasting, the enzymes are in a phosphorylated state. When the body is in a well-fed state, who is the key molecule that should come? The one and only one. Who is that? It is insulin. Okay. Now, this insulin has two actions. The number one action is it activates an enzyme called as phosphodiesterase. What is the action of phosphodiesterase? The action is it converts a cyclic AMP to 5' AMP. So, even if we just say AMP, it is 5' AMP only. So what is this 5' AMP? 5' AMP is adenosine monophosphate in which the phosphate group is attached to the 5' position. So even if I say AMP or 5' AMP, usually the phosphate group is attached to 5' itself. So it is not a separate thing from AMP. It is AMP only. We usually call it as AMP, but that is the same as 5' AMP. It is because of the position of the phosphate group. Okay. So what we see is the cyclic AMP is converted to 5' AMP. So what will happen to the cyclic AMP level? The 5' AMP level is increasing, and what we can see there is the cyclic AMP level is decreasing. So what will happen? The enzyme cyclic AMP dependent protein kinase is not getting the cyclic AMP now, which means it is inactive, which means the enzymes are not getting phosphorylated. Right? Now this insulin has one more action, and that is the insulin can activate an enzyme, and this enzyme is called phosphatase. What's the action of phosphatase? The phosphatase is the enzyme that take off the phosphate group from every protein. So whatever enzymes are phosphorylated is now getting into a state which is there where there is no phosphate group. Otherwise, dephosphorylation is happening to these enzymes, which means under the influence of insulin, whatever is getting phosphorylated is now becoming in a dephosphorylated state, and whatever new enzymes, they are not getting phosphorylated because that enzyme is not acting. Okay. So in short, so what we understand is that when the body is in a well-fed state under the influence of insulin, the enzymes are in a dephosphorylated state. Okay. So I hope that you understood the covalent modification being done by the hormones glucagon and insulin. Glucagon phosphorylate, insulin dephosphorylate. Okay. Now what we are going to learn is how we can apply this phosphorylation and dephosphorylation to various pathways that we'll explain with a small role play. So we have seen what happens in a well-fed and a fasting stages. Now we are going to apply this concept in various metabolic pathways. So if we want to apply it, I'll just tell you some concepts. Number one is whenever an enzyme or whichever enzymes are active under the influence of insulin is active in the dephosphorylated state. So the hormone of the well-fed state is insulin. Whatever is active is in the dephosphorylated state. Concept number one. Number two is that whatever enzymes are active in the fasting stage is under the influence of glucagon, and under the influence of glucagon, it is active in phosphorylated state. Simple two sentence that we have to learn. Now, to every pathway, we have to ask three questions. Okay, now finally we'll get the answer whether that pathway is active in phosphorylated or dephosphorylated. The three questions are number one is whether it is fed or fasting. The second question is this enzyme is active under the influence of insulin or glucagon. The third question is like we'll get the answer whether this is active in dephosphorylated or phosphorylated state. Okay, shall we start? Yes. Now, I'm going to give you a role. Now you are going to act as a glycolysis. Okay. So glycolysis means what? It is cutting of glucose, which means that this should happen when the blood is having glucose, right? The blood has glucose means well fed or fasting. It should be well fed. Okay, we are going to try that. So, first question I'm going to ask, that is, glycolysis is active in well fed or fasting. Okay, that is right. So, it is well fed. Fine. Now, let me ask you the next question. Is it under the influence of insulin or glucagon? Okay, so that is right, right? So it is under the influence of insulin. Now you have to answer the glycolysis pathway. The regulatory enzymes are active in phosphorylated or dephosphorylated. Yes, correct. So that is the answer. Okay, so we understood that glycolysis is active in dephosphorylated state. Now I'm going to give you the second rule. The second rule is like you are going to act like a gluconeogenesis. Gluconeogenesis means newly we are synthesizing glucose. Why? We don't have glucose. So I'm going to ask the three question to him. Well fed or fasting? Is it right? Yes, it is fasting. Okay. Now I'm going to ask the second question. Are you active? Do you love insulin or glucagon? Okay, correct. So it is glucagon. Now we are going to get the answer for the question whether it is active in phosphorylated or dephosphorylated. Okay. Yes, correct. So we got it like the enzymes of gluconeogenesis are active in phosphorylated state. Now I'm going to apply this to glycogen. Now we are going to act like glycogen synthesis. The regulatory enzyme is glycogen synthase. Okay, now he's taking the role of glycogen synthase. We'll ask the same question, fed or fasting. Glycogen is synthesized means we have enough glucose. That is why we are synthesizing glycogen. That is the clue that I'm giving you. Okay. Okay, right. So that is fed. Okay, once that is over, then I'm going to ask this question. Which do you love? Yes, it is. It should be insulin, right? Now, glycogen synthase is an enzyme active in the third question is this. Yes. So it is active in dephosphorylated state. Okay. Now I'm going to make you the opposite. Yeah. Now you are glycogenolysis. The regulatory enzyme is glycogen phosphorylase. Glycogenolysis means we don't have glucose, so we are pushing all the glucose out from the glycogen to the blood because we don't have glucose. That is the only clue that I'm giving you. Now I'll ask the first question. Is fed or fasting? Right, it is fasting. Okay. Now, the second question. Yes, very good. So, glycogen phosphorylase is an enzyme active in which state? Is it right? It is active in the glycogenolysis should takes place in the fasting state, right? So, it should be active in which state? Yes, right. That is the answer because glycogenolysis is under the influence of glucagon. So, under the influence of glucagon, the enzymes are active in which state? In the phosphorylated state. Okay. Now, we are going to jump to the lipid metabolism. Shall we? Okay. So, now you are going to act like fatty acid synthesis. The rate limiting enzyme is acetyl coenzyme carboxylase. So I'm going to ask the first question. Synthesis always happens when the body is in which state? So my first question is fed or fasting? Is it fasting? Like the fatty acid synthesis is in fasting? No, right? So fatty acid synthesis should be in which state? Fed state. Yes. Now he got the answer fed. Now I'm going to ask you which is the hormone that you like. Fed means it is insulin or glucagon. Yes, right. That is insulin. Okay. Now this enzyme, acetyl coenzyme carboxylase, is active in which state? Under the influence of insulin. Yes, correct. So, acetyl coenzyme carboxylase is active in the dephosphorylated state. Now, I'm going to give you the last role, okay? So, this is about cholesterol synthesis. Cholesterol synthesis means the body has enough glucose. From that, we are getting enough acetyl coenzyme. From there, cholesterol is being synthesized, which means the first question, fed or fasting? Yes, correct. Fed. So once you get this answer right, I'm going to ask the next question. Insulin or glucagon? Yes, he likes insulin. That means it is active in under the influence of insulin. Now, the rate limiting enzyme of cholesterol synthesis is HMG coenzyme reductase. It is active in which state? Dephosphorylated state. Is it right? Yes. So give him a good clap. So he had answered everything well. So this is how we can apply the concept of this three questionnaire and get the answer in which state each and every enzymes are active, phosphorylated or dephosphorylated. Is that clear? Yes. So we have learned what is the covalent modification and the hormonal regulation. Now I'm just moving on to the allosteric regulation. What is its concept? The concept of allosteric regulation is substrate favor forward reaction. This is otherwise called as the feed forward reaction. Okay. Now, if there is product accumulation, definitely we know that it is going to inhibit the forward reaction. So that is called feedback inhibition. Okay, now we are going to apply this concept to various enzymes, allosteric enzymes. The first example that I have given here is phosphofructokinase. Phosphofructokinase is an enzyme which convert fructose 6-phosphate to fructose 1,6 bisphosphate. Okay, so that is a role. Now I have given here various activators as well as inhibitors. Just we have to check whether the compound that is given there is substrate or product. Then that will give you the answer whether it is an activator or an inhibitor. So I'm taking the example of 5' AMP. What is 5' AMP? Like in the case of glycolysis, we know that the glucose is converted to glucose 6-phosphate, then finally converted to pyruvate. Now, this pyruvate, during this process, we know that NADH is produced. This NADH is entering into the electron transport chain where we know that ADP is converted to ATP, right? Now, when ADP is converted to ATP, from where is this ADP coming from? It should be from AMP. Okay. So, during the process of glycolysis, that is the phosphofructokinase is an enzyme of glycolysis. Okay. The function of glycolysis is to, final function is to generate ATP. The ATP is formed from ADP. ADP which in turn is formed from AMP. AMP is, even if I write AMP, it is 5' AMP only. So, everything is formed from, like, this ATP has to be synthesized from ADP, which in turn has to be synthesized from AMP. So, my question is, what is the role of AMP in this step? Or in glycolysis, right? So in glycolysis, it is acting as a substrate, right? So that is why we are going to learn this is a substrate. So just see, it is an activator here. Now the next one, that is fructose 6-phosphate. What is the role of fructose 6-phosphate here? I've already told you that this is the one which convert fructose 6-phosphate to fructose 1,6 bisphosphate, right? So what is fructose 6-phosphate here? It is a substrate, right? So this is a substrate. Hence, it is an activator. Okay. Now we go to the other side. ATP. Already I have told you the function of glycolysis is to produce ATP. So it is a product. So as it is a product, it is an inhibitor. Very simple. Now low pH. How low pH is acting as an inhibitor? I'll show you. Glucose becomes pyruvate, and this pyruvate if there is no oxygen or in anaerobic condition, that is where the pyruvate can be converted to lactate, right? So it is lactic acid, which means this is low pH. This is like decreased pH, right? So what is decreased pH for this glycolysis? It is a product. So you can see this is a product. Hence it is an inhibitor. Now go to citrate. What is citrate for glycolysis? The citrate is like, we know in glycolysis pyruvate is formed. From pyruvate we'll get acetyl coenzyme A. This combines with oxaloacetate to produce what is called citrate. Thereby it is entering into tricarboxylic acid cycle. We know that. So this citrate is coming after glycolysis in the product side, right? So citrate is a product and hence it is an inhibitor. So whatever compound you are getting, you just think whether it is a substrate or a product. Then you can very well answer whether it is an activator or an inhibitor. Now we will take the next example, acetyl coenzyme carboxylase. So before going to about its activator or inhibitor, we'll see what is the role of this acetyl coenzyme carboxylase. The function of acetyl coenzyme carboxylase is to convert acetyl coenzyme to malonyl coenzyme. Okay. Now where is this coming? This is coming in fatty acid synthesis. Okay. Now from where do we get this acetyl coenzyme? Like this acetyl coenzyme is actually inside the mitochondria. Now this mitochondria, inside the mitochondria, this acetyl coenzyme is transported out of the mitochondria as citrate. Okay, it is transported as citrate. Okay, now this citrate later on becomes acetyl coenzyme and then it is getting converted to malonyl coenzyme. All these things you will learn later also. Just to make you understand the concept, I am saying all these things. Okay, fine. Now, if this much I am giving you, because we have not yet started the metabolism, I am just giving you what is the role of all these things. Okay, so this is what is the role of citrate here. So first you see the citrate. This citrate is coming before this acetyl coenzyme carboxylase, right? Because citrate is the one which is giving off this acetyl coenzyme. That is, acetyl coenzyme is transported as citrate. So when the citrate is coming, it is going to activate this enzyme because more and more citrate is there, which is coming actually in the substrate side. So this is a substrate and hence it is an activator. Now go to the other side, malonyl coenzyme. So that is very simple because it is a product. Okay, now what about this acyl coenzyme? What is acyl coenzyme? It is a fatty acid, right? The fatty acid, when it is combining with coenzyme, it is called the fatty acyl coenzyme or simply acyl coenzyme. Okay, so what is acyl coenzyme to this enzyme? Is it in the substrate or in the product? It should be in the product. Okay, so this is a product. You see, it is an inhibitor. Like that, if I give you one more example, ALA synthase. So I'm writing here ALA synthase. This enzyme has an inhibitor and that is ALA synthase inhibitor is heme. Why? Because ALA synthase, the function is to produce heme. So heme is acting as an inhibitor of ALA synthase. So this is a very simple way to learn allosteric activation and inhibition. Just see whether it is a substrate or a product. If it is a substrate, it is an activator. If it's a product, it is an inhibitor. So that is about the concept of enzyme regulation.